Okay. Thanks for coming. Thanks for, I think there are people online. Thanks for being here online. So it is another session of the Molecular and Developmental Biology series. And we have the pleasure of having someone who is very familiar, I think, to the vast majority of us in CEFISES, but who has not been to the Villeneuve before. So it is nice to get a good reason to have a first invite to Maël Lemoine, philosopher of biology and medicine. He's been in Bordeaux now for quite some time. Well, the floor is yours. Without further ado, take it away.
Thank you very much, Charles. So thank you for the invitation. I'm of course very happy to be here among you today and to share some of my thoughts about aging. So I would like to start with a general question. So why studying aging? So in fact, just to give you a few reasons why, because it's not a topic that is that big in philosophy of biology in particular, or even philosophy of medicine for that matter. So the first reason I think is that first reason for me is that aging is obviously a major constraint on organisms in evolution, and this is one that is understudied, I think, but it's a major constraint. On the other hand, there is a so-called evolutionary theory of aging, which is not in fact very developed. It's rather old. There are, of course, recent developments, and I think the main consensus among biologists who are studying aging is that it's not very helpful. It's very abstract. Everyone agrees with it, so it's good. It's not bad, but it's not very developed. So it's not, we would not be able to use it very much to get some information. Now, very recently, there is really a boom in comparative and phylogenetic approaches to aging. So people are studying aging across species, and we have, of course, a lot of data coming on that, and discovering also at the same time that some organisms have very, very, very weird, unexpected patterns of aging. Which is also very surprising. So some of them, for instance, would be non-senescent, so do not age at all, or they do not have anything that looks like aging, and others would have weird patterns like even what is called negative senescence, for instance, meaning that the older they get, the less chances they have of dying. So this is also very counterintuitive for people that are focusing on organisms like ours, of course. Mostly and mainly the personal reason why I would be studying aging is that I think it would be very helpful if we could at least go some steps towards a theory of stages in the evolution of aging. So this is one thing that I have developed in that paper. So of course you cannot read what's on the figure, so I'll just give a few explanations. It's a review paper where I in fact gathered all the information we have on, let's say, what could have been the very first stages of the evolution of aging. So I will not develop that, but give or take, the idea is that aging is something that appeared rather late in evolution, probably was clear only with the appearance of bilaterians. And you have, nonetheless, some of the mechanisms of aging, they have appeared before, but they were not mechanisms of aging during earlier stages of evolution. So that's one thing I'm very much interested in, and the talk I will give will be, let's say, in that context. So we'll focus on a specific question, but the talk will be in that context. And the other and last reason is regarding the book that I know some of you have read, and I'm grateful for that, is that I think that it's also interesting to study aging as probably a common pathway to many age-related diseases, or maybe a common pathway inside of these age-related diseases. So that's also one thing that is interesting and is very active in biomedical science now. People are studying the mechanisms of aging as a risk factor for cancer, for instance, or Alzheimer's disease, a separate process, but one that would contribute to these diseases. So that's why I'm also interested in that. Okay, more specifically, the question I will raise today is what I call cell aging reductionism. So this is what I want to investigate with you. So let's start with a very general claim, one that I think is pretty uncontroversial, which is that all biological entities, from DNA and proteins to organisms and possibly beyond, degrade with time. So we'll just take a very broad term, very broad notion, because degradation is a controversial term, in fact, but everyone agrees that these entities degrade with time. And aging is one specific form of degradation among many. And generally, this specific form of degradation is contrasted with other forms of degradation, such as injuries, intoxication, infection, and so on. More specifically, and this is not to be taken as a definition, properly speaking, of aging, but just as a contrasting feature of aging with other forms of degradation, it's thought of as a form of degradation that is necessary or intrinsic or ineluctable. So that's of course, very rough approximation, but it will be necessary, obviously, and I think sufficient for my talk here. But of course, it's a much bigger question to distinguish aging from other forms of degradation, or even to define degradation, of course. So now, if I focus a little more on the next concept that counts is this idea of cell aging. So cell aging is, or the idea that cells are aging, means that there is a kind of cell autonomous, so this is the term that biologists would often take. Sometimes they say cell intrinsic. Definition is not completely clear, but it means that it's something that probably happens inside of the cell membrane, and also that would happen whatever the context the cell is in would be. Okay, so whatever the context of the cell, this would happen anyway. So it's a process of degradation that necessarily results from the normal functioning of cells, although it can, of course, be modulated by so-called non-cell autonomous factors. So the factors that would be outside of the cell. So for instance, I don't know, something that is toxic to the cell could accelerate the aging of the cell, but anyway, the aging of the cell is a process that will take place. Okay, so the idea is that you have something, a process inside of a cell, and this process is degrading the cell. This is what I would call cell aging. Okay, simple idea. So, this comes from a paper very far away. I don't think you can read from there, but I will explain. So this comes from a paper, very famous paper, in fact, second version of a very famous paper, called the Hallmarks of Aging. And this paper is listing the various mechanisms of aging we know of. And so what you can see here is that, so there are several mechanisms of aging, ranging from the loss of proteostasis, meaning the loss of the balance between protein degradation and protein synthesis inside of a cell, loss of proteostasis to dysbiosis, which is something that is more systemic. So dysbiosis is the perturbation of the microbiota in an organism. So these are the mechanisms of aging. And here, the ones that so this is my figure here. I mean, the orange part of the figure is not from the original paper. These are cell aging mechanisms. So they are represented here at the bottom because it's a level figure, like you have meta-organisms here, molecules here. Okay, so these three things are happening inside of cells. And beyond that, it's less clear that it's inside of cells. It's something that is more relevant, let's say more related to organisms. So what I call cell aging reductionism, this is my phrase, so it's not something you would find in the literature, means that aging at other levels here would originate in the aging of cells. So if I recapitulate the idea, it's a very simple idea. Cells age, and because they do, organisms would age. Okay, so that's what I call cell aging reductionism. This is just one quotation just to show you that it's not a straw man argument, that this is a position you would find very commonly mentioned in the literature. At least 10 years ago it was the case. I would probably qualify that today, but it's still a position that exists in the field. You have this quotation from the same paper, but 10 years ago, well, a little more than 10 years ago. The time-dependent accumulation of cellular damage is widely considered to be the general cause of aging. So that's very clear. Okay? Accumulation of cell damage is the general cause of aging. And these mechanisms of aging, those that are in the orange box here, are called by Lopez-Otin and colleagues, are called the primary hallmarks of aging, which is also quite clear. Okay, if you look at the definition, they say, in the 2013 paper, they say that these mechanisms are triggering aging. Okay, so this is again, in my definition, where aging originates. So now if you get the idea, I would like to introduce a distinction that I think will, just to avoid some confusion that everyone would make, so I think it's important and useful here. I'm distinguishing cell aging reductionism from cell reductionism in aging research. Okay, so what do I mean by this? I mean simply that there is, and it's quite common in biology, of course, it's to look at things from the perspective of cells. As you can study what aging at the level of cells. It's not the same position as to say that the aging of organisms causally originates in the aging of cells. What it means is that you can look at the degradation of an organism at the level of its cells. So it could be the effect of aging. I mean, if cells are degrading, maybe it's the effect of aging. Doesn't matter. Okay? So it's very different. It means that you can study aging at other levels and you can interpret it, for instance, as actions of cells, which is not, again, the same thing. Cells can act on an organism, on the overall organization of an organism, and yet not due to their degradation. So this is very distinct. So that general position is something you can find in, for instance, Nicholson's paper in 2010. The paper is called Biological Atomism and Cell Theory. So that's a paper on cell reductionism. Okay, so what I'm talking about is not cell reductionism. Okay, it's something beyond that. Okay, so now let's go a little more into the detail. So first point, here I want to define a little more precisely cell aging reductionism. So I'm starting from this assumption that some composed entities age and die strictly because their components do. Okay, so if let's take another physical object. Let's take the example of a bridge. Well, why would a bridge collapse? Simply because the stones or the mortar or whatever the bridge is made of would degrade, and then at some point the physical organization would not be possible anymore and the bridge would collapse. Okay, so it's a very simple example. And the question is, are organisms like that? Is it what's happening to organisms as well? In fact, there's an important difference between organisms and other physical objects. And this difference is, or can be called, or sometimes called, renewal or replacement or repair. So if you think of a bridge, you can replace the stones or the mortar or whatever. You can do that. And organisms, as you know, most of them at least do that, at least to some extent. So this makes a huge difference. Because of course, if you replace parts, then the aging of the entity would not depend necessarily or directly on the aging of its parts. Okay, so that's a very simple idea. So here is one example where the aging of the entity, let's say the organism here, strictly depends on the aging of its parts. So this example is C. elegans, Cenorhabditis elegans, the small worm you can see here. This is an organism that is called post-mitotic, meaning that its cells would not divide after maturity. So it has some repair mechanisms, but they are very limited. So in a sense, here, the aging of the organism directly results from the aging of the cells of the organism. Because again, the organism has no means of replacing its degraded cells. So very simple idea. Here, that's the other extreme. It's the hydra. So the hydra is a small organism as well, where that is supposed to be non-senescent, so it doesn't age, meaning that you don't see any degradation at the level of the organism. But of course, individual cells degrade in that organism. Okay? So you have the two opposite examples. So very simple example. So here, C. elegans, the aging of C. elegans would probably directly depend on the aging of its component cells. Here, in the hydra, even though cells are degrading, the organism is not degrading. Okay, so cells are replaced so fast and so well that the organism would not degrade. Doesn't mean that it cannot degrade, of course. It means that it can not degrade, which is different.
Mm-hmm.
Okay, so if you have this distinction in mind, you can understand that idea that I call metacellular. I'm not the only one to call it that way, but I use the phrase metacellular aging. So the idea that multicellular organisms with self-renewing components would age if and only if the aging of the components leaves a trace, leaves a cumulative trace of degradation after they are gone, which does not happen in the case of the hydra. Okay? So cells can degrade, and the question is whether this degradation would leave a trace, a lasting trace. So after the cells have disappeared, you have something that's left and would change the organization of the organism. Okay, so this is in fact the right formulation of cell aging reductionism. So cell aging reductionism, if I summarize the idea, is not the idea that organisms age just like a bridge does, like stones that would never be repaired, let's say, like stones degrade and therefore the bridge would collapse at some point. It's not that idea. It's rather the idea that when they do that, the organism can never be restored as good as new. Okay, so it's a default in, you could say that, it's a default in repair or renewal or whatever you want to call that. Okay, so that's the idea. So just to make it a little more concrete, biologically speaking, because this is very abstract said like that, so in senescent organisms, for instance, cell aging would leave a trace in the cell lineage. So cell lineage, meaning that, as you know, cells would divide. This is the way they are renewed. And it's possible that you would see the degradation not in one individual cell, but in a lineage of cells. That is one cell and then the daughter cells, if you want, so to speak, you would see something that is accumulating over generations. Okay, so it's not an individual cell that is aging, it's a lineage of cells that is degrading. So that's the first idea, so that's a little more concrete. And there are also other possibilities. Another one is that whatever is not cellular in an organism, and the main example is called, for instance, the extracellular matrix. Okay, so by definition, something that is not inside of the cell, that is not cellular. The extracellular structure would be changed and altered by the behavior of some cells. And this would last even after these cells are gone. So the extracellular matrix, you can think of it as a kind of very complex net around cells that would put them together and organize them physically and spatially And this structure is produced by cells themselves. And after they're gone, of course, after they are replaced, for instance, by another cell, this structure remains. Okay, so you have traces of their past activities, so to speak. So obviously, in non-senescent organisms, on the other hand, cells apparently do not leave, cell aging rather, do not leave any of those traces. So the lineage seems to be constant. Cell lineage still seems to be constant, and the extracellular structure would also be constant. Okay, just to be more concrete. So the view I want to defend here is precisely the opposite. So this is what I will try to defend here. I will give you 3 arguments in favor of what can be called then cell aging anti-reductionism, or 3 arguments against, if you prefer, cell aging reductionism. And the idea is that, very precisely, is that other components than cells necessarily leave a cumulative trace as well. In other words, not everything in aging comes from the cumulative trace of the degradation of cells. That's the idea I want to defend. So, of course, my point is not to say that the degradation of cells has nothing to do with the aging of the organism. It would be a weird position. But simply to say that there are important processes outside of the degradation of cells that would result as well in the aging of the organism. So here are a few examples of those things, as you can find them in the literature. So the degradation of the ECM, extracellular matrix, that I just mentioned, is something that is to some extent independent from what cells are doing and how cells degrade. There is also probably a natural evolution of the microbiota in some organisms that is also probably to some extent autonomous and does not depend on the degradation of the organism or the cells of the host, let's say. And there's also some theories and also facts about a kind of progressive noise that is not to be attributed to any individual cell and should not be considered to originate in damage, properly speaking, but this noise would be also would accumulate. So it's progressive noise produced by normal cells, let's say, and would progressively disturb their communication, let's say. So these are 3 just examples or ideas that you find in the literature about that. And of course, I mean, people who would defend this position of cell aging reductionism, like Lopez-Otin and colleagues that I just mentioned, they claim that these non-cellular components of aging that leave a trace do that only because cells themselves age. Okay. So for instance, just to take a few examples, you can find in Lopez-Otin this idea that the microbiota is degrading in an organism. If you think of the human organism and the gut microbiota, for instance, it degrades in an organism only because the gut itself has already degraded, only because the cells of the gut are already degraded. So this is the kind of claim they would make. So they would say, yeah, of course, the microbiota would have an impact on the aging of the organism, but it's only because the organism is already old that the microbiota would degrade it further. So it's a requirement. You need an old gut to have an old microbiota, so to speak. Okay, so which is kind of very weird. I mean, it's a very biased interpretation. It's very possible, but it's very biased. It's a view that is, I mean, it's probably a strong ideological view, or at least it's strongly biased in terms of a theory of cell aging. Or they would say, that's also typical, that that's a debate in the literature, in fact. We don't know whether the extracellular matrix is degrading on its own, or whether it's because fibroblasts, that are the cells that are producing this structure, are aging and degrading. So that's in fact a big problem for, especially in some diseases where you have problems in this thing like fibrosis, for instance, is a pathological phenomenon when you have an overgrowth of the extracellular matrix. And there, people don't know whether it's because fibroblasts are dysfunctional or whether they are dysfunctional because the extracellular matrix has changed its structure. So there is a debate here in the field. Okay, so let me can drop the last idea. Okay, good. So do you have questions on that? And then I will give you the 3 arguments I want to develop in favor of cell aging anti-reductionism. But maybe you have clarification questions, short ones at this point. Yeah.
Can I? Thank you. No, I was asking because you said something concerning the traces, not refer to cells, but what are the traces?
They could be in cells. You would find them in cells. Yeah, but the thing is that the cause of the degradation is not here anymore. So you cannot say, okay, that cell from generation, I don't know, N, for instance, this cell has had, for instance, some mutation that happened. Okay, so the hit is here. The idea is that the hit that happened here happened to a cell that has now disappeared. Now that we are at generation N plus whatever, for instance, you would not see that cell anymore. But that thing that happened has left a trace, here in my example, in the other cells that came from that. So the mutation is conserved.
So, in that case, the trace is the mutation.
Yeah, in that case, it's a mutation.
I was thinking about which are the kind of traces that you are referring to concerning cells. So mutation is one example.
Oh, yeah, so that's one of them. And that one would be found in cells, but others would not be found in cells. So for instance, for the extracellular matrix, which is the example I took, if you look, I think I have a picture later, so you will see how fibrosis looks like, but it's a macroscopic picture, so you would see how it looks like at the level of the tissue, but at the microscopic level, what you would see is that you have a structure that is, in fact, well balanced like this. It looks like a net. And when you get older, in fact, it does this. Like it looks more like some fibers that are all in the same direction. And it becomes thicker, harder, and stiffer. Okay, so it's less, I mean, it's less good regarding many biological functions. For instance, the immune system or cells of the immune system cannot go through it as easily. Macrophages and other cells, they would not go as easily through the extracellular matrix. So that's the kind of traces I mentioned here. Okay?
Thank you.
Yeah. So 3 arguments. So they are very simple. In fact, the first one is simply to say that, okay, so I should decompose that. The 3 arguments would rely on 3 different stages of the evolution of bilaterians in general. So animals, I'm focusing on animals here, and bilaterians more specifically. So the first stage is about the emergence of multicellular organisms. So that's the first stage I'm considering here. And here, what I want to say is that the aging of cells is not necessary. No. The first argument is it's not sufficient. The aging of cells is not sufficient to have an organism aging. So we'll take one example. And what I want to focus on here is that what is necessary is a certain cell population dynamics. Okay, so that's the that's in fact, this is what is sufficient. Well, at least here I will defend the fact that it's necessary as well. Second argument, so that's the second stage or later stage of evolution. Some metazoans emerged that were senescent. And what I would like to show here is that even if cells did not age, some multicellular organisms could age. So it's not even necessary that cells age. Okay? So it's not sufficient. That's my first argument. My second argument is that it's not even necessary that cells age. Why? Because I will focus on the idea that cells can produce perturbations in the multicellular entity, even when they are not degraded. And then the third argument I want to make is that when you take specific, so it's the let's say the last stage of evolution I will focus on here, the emergence of and the variations, let's say, among the various branches of the tree of bilaterian life, if you want. So in bilaterians, so further stage of evolution towards animals like us, typically. Even if you think that there are specific constraints on those organisms, that is, you would say, okay, they have all kind of the same cell population dynamics, mechanisms of repair, more or less, and all that, then some people can think, in fact, that it's the modulation of cell senescence, or the aging of cells, if you want, that would explain the variations in the rate of aging across these organisms. Some organisms, as you know, would live like even 3 days or even 2 days, and others can live for centuries. So what's the difference? Some people say, well, the explanation is in cells themselves, the way cells last, let's say. Okay, and I will defend that in fact it's probably not the case. So it's not what explains these variations. Even if you think that the cell population dynamics is the same, or roughly the same. Okay, so these are my 3 arguments. So I will go into them into more detail, and I will try to make them short though. Okay, so the first argument, so here you have a very rough representation of the tree, at least the branch of the tree that leads to bilaterian here, very simple, and this is the step at which I'm focusing on here, emergence of multicellular animals. So here my argument in a nutshell is that cell population dynamics and not aging cells alone would make multicellular organisms age. Okay, so to do that I will go into 2 arguments by August Weismann, who is a German biologist from the 19th century, who made a very, very influential, well, made these 2 very influential arguments. And I think it's important to start from there. I'm not much of a historian here, so sorry in advance if I made some oversimplification, but I think these arguments are still very influential today, under a simplified form probably. So suppose now that you have a generation of single-cell organisms. Okay, so bacteria, for instance, or yeast, whatever. Okay, so you take a generation of them into a population. So meaning a generation, okay, all born roughly at the same time. Suppose now that all of them are degraded for whatever, physical reasons, whatever. Suppose that all of them are degrading. And now, suppose that the degradation is passed on to future generations of cells in that population. When the cells are dividing, the damage would be passed on. You cannot get rid of that. They cannot get rid of that damage. They cannot repair or whatever, so they pass that damage on to the next generation. Consequence, and that's Weismann's first argument, the population would go extinct at some point. Okay, so you can represent it in a simplified way like this. So if, let's say, the lighter the color, the higher the level of degradation, and if we follow just one line here, you see that you had, okay, let's say, perfectly working organism here, cell division with damage here, and damage would accumulate over, in fact, in all branches. So the whole population would go extinct. So that's a very simple argument. So, in fact, if we can still observe unicellular organisms today, and that's what Weissman is concluding, it means that cell-autonomous aging is impossible in single-cell organisms that are inheriting degradation, that would inherit degradation. So there must be something that, you know, get rid of the degradation, because otherwise they would go extinct. So his solution was simply to state that unicellular organisms cannot age. Okay, so that's the first dogma that Weissman proposed, just to solve that problem. Unicellular organisms cannot age, and there are still many biologists that are convinced that unicellular organisms cannot age, because if they did, again, they would have disappeared. So here's Weissman's second argument. So now we are not talking about single-cell organisms anymore, but multicellular organisms. And we are thinking of multicellular organisms that reproduce not sexually, but clonally. Okay? So they would divide as well, or they would produce one out of another without any sexual reproduction. Well, that's the same thing here, in fact. If cells age and inherit damage, then multicellular organisms would also go extinct, exactly for the same reasons, of course. But, of course, as we know, population of multicellular organisms have not gone extinct. So, and we also know that multicellular organisms do age. At least we know many of them that do. Okay, so how can we explain that? Weissman's solution is that, first, cell-autonomous processes of aging would cause inherited damage in what he calls somatic cells. So he's the one to introduce this distinction between germline cells and somatic cells. Okay, so damage accumulates in lineage of somatic cells. But there's no cell-autonomous processes of aging in germ cells, germline cells. That way, you can preserve the lineage of organisms. But at the same time, you would see individual organisms age. Okay, so his solution is simply to say that we have two types of cells. And this is represented like this, if you want. If you look at one lineage, so this represents, in fact, cells. You would see here, let's say if you take that one, you would see that's one organism born here, and you would see the somatic cells that would go extinct progressively while the germline is preserved. And at some point later in life, for instance, you have an event of reproduction. Okay, so that's roughly Weissman's idea to solve the problem of aging. So this distinction, germline soma, was introduced first in the context of aging, to understand how you can still have organisms today, although organisms age. And if I summarize the two arguments, his solution is, first, unicellular organisms don't age, they cannot age. And second, germline cells don't age while somatic cells do. So if you take that all together, the argument is that aging comes from, or is something that concerns only somatic cells in a multicellular organism. That's the basic idea. Okay, so we have similar solutions to the same problem in the so-called disposable soma theory proposed by Kirkwood more recently. It's not that recent, but more recently. So now we probably, of course, want to challenge those arguments. So first, it's something that probably, at least biologists among you know, that cell-autonomous aging probably exists, in fact, in all unicellular organisms. So we know some of them where it does, for instance, yeast. But probably now we know that probably in all unicellular organisms there is something like progressive necessary intrinsic degradation. One sure sign of that is that the reproductive value decreases with time. At the same time, there is a problem with clonal multicellular organisms. So these organisms have no clear, the problem is that you cannot find any clear germline soma distinction in them. Okay, meaning that any cell, basically, if it's separated from the organism, can become an individual organism in its entirety. Okay, so you don't have any division of functions that is that clear in many of them. It's the case for sponges, corals, and even the hydra. Okay, so it's not really one cell that can become a full organism, but it's generally a limited number of them. Like you can have, if you have one of every cell type, you would have that. So in the hydra, for instance, almost all the cells are replicative. They are considered to be in fact stem cells that can give birth to any type of cell in the hydra. And at the same time, and at the third paradox, if you look at those organisms, as I said, I gave the example of the hydra before, in fact, individual cells in those organisms, they do age as well. So that's even more paradoxical. Yet, of course, populations of unicellular or clonal organisms do not go extinct, or they have not, at least. So how can we solve that? So the only solution to that problem, I think, comes from the cell population dynamics. So let me explain. So we've known for a long time now that cell division is, in some organisms, asymmetric. So the example is the yeast. The classical example is the yeast, where you have, when the yeast is reproducing, you would have what is called budding. That is, you would see a small cell coming out of a mature cell. So you can distinguish the mother cell and the daughter cell. Okay, the daughter cell is much smaller than the mother cell. So you have so-called asymmetric division. And what we know is that these cells can do that for a limited number of times, after which they cannot reproduce anymore and would die. Okay, while the daughter cell is as good as new. So it's very intuitive for us. It's like a mother and daughter, and the mother is accumulating damage. It's older, it will die sooner, and the daughter cell is like reinitialized somehow, although it came from cell division. Okay, so that's asymmetric cell division. So Kirkwood at some point argued that it's probably the equivalent of the germline-soma distinction, something like this. Okay, so you would have inside of the same of the mother cell, you would have something like the germline, and the germline is the daughter cell, what becomes the daughter cell, and the soma is the rest. Okay, so you have that kind of distinction, and he says, so you can save Weissmann's argument. In this case, you can say, well, it holds. We have this soma-germline distinction, and that's why these unicellular organisms are aging. Okay, so Weissmann's theory is saved somehow. But of course, it's wrong. It's wrong, why? Because both cells can divide, so it's weird to say that you have a germline and a soma. What is the soma exactly? Is it the whole cell or something inside of the cell? And if it's something inside of the cell, what exactly? And the two lineages are degrading. Like the daughter cell would degrade. I mean, what will come out of the daughter cell will also degrade. So the distinction is not really clear here. You cannot really make that argument. And on top of that, biologists have discovered more recently that there is another case where you would not see a morphological asymmetry, like you have a bigger mother cell and a smaller daughter cell, but you can have functional asymmetry. Like you have cell division in E. coli, for instance, which is a bacteria. You would see when they divide, you have also asymmetry, but not in the size, not in the shape, but in the level of damage. Okay, out of two bacteria, when you have two bacteria born of the same one by division, one of them has more damage than the other. Okay, so it's called functional asymmetry. Why functional? Because you would see that because one of them would simply reproduce less in the long run than the other one. And also because you would see aggregated proteins, but that's another question. So think of that. The figure comes from this paper, the paper where functional asymmetry was discovered. What they modeled, so they modeled here generations coming from the same let's say, original cell. And here, what you can see is the time before the cell divides. So you can measure the time before the cell divides. And what you can see here is progressively you have this asymmetry that is appearing. So it means if you look here, for instance, it means that from the two cells that divided from the same event here, cell division, you would see one that would take longer to be able to divide again. And if you follow the lineage that is always taking longer, in the end you would see one cell that just cannot divide anymore. Okay? So this means that if you take a whole lineage, you see the phenomenon of aging. Okay? So it's not the cell, properly speaking, that is aging. But if you compare generations in the same lineage, you would see progressive degradation. So something is accumulating. And what is interesting is that this accumulation is asymmetric. Okay? So if you think of that, if it's asymmetric, what is asymmetric is the distribution of damage. So simply think, for instance, if you have one cell, it has accumulated a certain level of degradation. Then the cell is dividing. There are two daughter cells now. One of them has all the damage and the other one has no damage. What happens? The one that has all the damage can divide, but it will be less efficient, probably, while the other one is as good as new. So if you have that, then of course, the population will not disappear. So what's the explanation? It's not the aging of the individual cells. The key phenomenon is the asymmetry. So that, which is one property of the cell population dynamics. Why I'm saying that? Because it's not just, I mean, the condition is not that you have one that is as good as new and the other one that is taking all the damage. The thing you need is simply that you have asymmetric distribution of damage. If you have asymmetric distribution of damage in the long run, you can have perpetuation of the species. This is the only condition you need. Okay? So that's why I'm saying that this is what you need. If you don't have, this is what you need for the organism to be able to exist. But on the contrary, if an organism is going extinct somehow or is simply damaging over or beyond repair, it means that this condition is not met. So this is what we need for an organism to age, a multicellular organism to age. Okay, so this...
I have a quick question.
It's
about bacteria or?
Yeah, this one is about bacteria. Yeah, this one is about bacteria. But you can see it's easily generalized, of course. I mean, it's just the dynamics. So that's what is important. I mean, the discovery is important. But what is, I think, even more important is simply to have shown the logics of it. For me, it's really a major paper in biogerontology. This is not a paper that is very often cited, but I think it's a major discovery. I mean, everyone agrees with that paper, but I'm not sure that everyone has seen, in fact, the consequences of that paper. I think it's really underestimated somehow. Okay. So, conclusion for that argument, cell aging is not sufficient to explain the aging of multicellular organisms. What you need is to specify the cell population dynamics. You need to know that to know whether an organism will be aging or not. So that's my first argument. So the second one is probably a bit more, I mean, it's easy to understand, but it's a bit more maybe paradoxical somehow. So I can simplify the argument by saying that what I want to show is that some multicellular organisms would age even if their cells did not. Okay? So it's more abstract argument because here I will try to find examples as convincing as I can, but I have no example of that kind of organisms where cells would not age. What I want to show is that it's not necessary. That did. Okay, so suppose that cells can have two different effects. Or you can observe, let's say, two different things in a cell. The first one is that the cell is degrading. And the second one is that the cell is producing perturbation. So degradation, the cell is self-degrading. That's one thing. The second one is that the cell is perturbing the multicellular entity. It's doing something that is producing some perturbation of, say, for instance, communication between cells, whatever. I think you can see immediately that it's not the same thing. You don't need, a cell doesn't need to be degraded to produce something that is perturbing other cells. Okay? The only thing, the simple idea is simply to say that you should not assume that a cell is a perfectly social entity inside of an organism. That everything it does normally should align with the goals of the multicellular entity. What we need is that it's doing, let's say, altruistic, it has an altruistic behavior long enough and at a sufficient level for the organism to survive and reproduce. That's the only thing we need. We don't need to think that all that a normal cell would do would be in the favor of the multicellular organism. So it means that they can perturb this organization. Okay, so that's degrading cell, perturbing cell, and you can understand that it's not the same thing. Okay, so that's the picture. I'm trying to go a little faster because I see that time is passing. I want to give the third argument. So that's fibrosis, progressive fibrosis. Okay, so that you can see here. So that's a young organism. I think it's a human, it's humans. Biopsy samples, and I don't know what tissue it is. I think it's probably skin here. I'm not sure. Okay, so you can see here, so that these are cells. You cannot really see the extracellular matrix here, but it's there between cells and around cells. And here you can see it increase simply because you have more of it. And then this is a 74-year-old person, and you can see here the extracellular matrix taking the surface progressively. Okay, so you have, so something is happening here. So what I'm saying is that these, inside of that tissue you have some cells that are producing the extracellular matrix, fibroblasts, and they do that on the basis of some physical properties of the extracellular matrix. Meaning that if you have less of it, they will produce more of it to compensate. Okay? This is called repair. Okay? But at some point you can see this balance is progressively, I could even say collapsing. Okay, so it's producing more and more of it, and the extracellular matrix is stiffer, and because it's stiffer, fibroblasts would interpret the signal as, okay, everything's fine, I don't need to produce more cells or something like this. And you would have this balance progressively disturbed. This is probably due to, you can take a cellular perspective and say, for instance, this is because of the cells. They don't do the thing they should do. Well, in fact, there's nothing wrong with those cells. They are perfectly functional. You can take some of them and put them in a younger organism. They would not disturb the younger organism. They would do exactly what younger cells are doing. It's just that they are doing that in a context that is, or in an environment that is progressively shifting or drifting towards something that is less efficient. That's all. But that's produced by cells themselves. Okay, so that's what I want to say here. So if you have that, my second argument is that this is sufficient to have aging. So you don't need cells to degrade, you just need them to perturb the kind of collaborations that cells have in a multicellular entity. This is what you need. This is all you need, in fact. So I'm not saying that this is all there is. I'm just saying that this is all that is necessary. You just need that. You don't need cell aging. But you can have, of course, cell aging on top of that. Okay, there are other examples of that. One that is called, there's a theory, a recent theory in aging, which is called the hyperfunction theory of aging. The idea is that, in that theory, the idea is that there is a certain pathway in cells that would lead them to, let's say, overeat nutrients and overproduce. Okay, so overgrow and all that. The idea is that this toll pathway is very good in a multicellular organism because it's helping it develop in early stages of development. Okay, so it's very efficient. you would probably produce tissue much faster thanks to that, because you can synthesize proteins much faster thanks to that pathway. The idea of the hyperfunction theory of aging is that you cannot, or selection could not have, natural selection could not have selected something that would, just switch the mechanism off once the organism has reached its maturity size. Because it's probably too late. I won't go into the details of that, but this is the basic idea. Okay? They say that's the cause of aging. And many of those who are defending that would also defend a cell reductionistic view of aging. And that, I think, is not consistent. Because I don't see why you would say that a cell that is still working on the tau pathway is dysfunctional or degraded. It's not degraded. It's simply functioning in an environment where you have a lot of nutrients and it's going to grow. So you don't have any program to simply switch off that. When you have the nutrients, you use them. That's all. But it's not good for the multicellular entity. Okay? So you don't need to say that cells that are constantly hyperactivated like this are damaged or even dysfunctional. They are neither damaged nor dysfunctional. But that could be an explanation of aging. I'm not defending that view particularly. It doesn't matter for me here. It's just to say that we should not say or think or suppose that it's a mechanism of cell aging. Okay. So don't think I need to go into the details of that, but just for the record, very quickly, I told you already in the beginning that Lopez-Otin and colleagues call these cellular processes the primary hallmarks of aging. And I should probably mention that the other hallmarks are called antagonistic hallmarks. And the idea here is that it's the responses of the multicellular organism to the damage that is accumulating. So these are mechanisms that would, in fact, they originate from mechanisms of repair, or mechanisms of protection against the bad consequences of the first ones, of the first mechanisms. These mechanisms would work for some time, and then the effect would be reversed. They would aggravate aging, just like the tau function or the hyperfunction. You have antagonistic pleiotropic effects. So it's good early in life, bad late in life. So typically, for instance, cell senescence is supposed to be good early in life. So generally, let's say the common wisdom is that cell senescence would protect an organism against the risk of cancer. So the cell would not be able to replicate anymore, and that's protective against cancer. But then, if these cells accumulate in a multicellular organism, they would produce something that is bad for this organism. Because these cells are not dead, they're still here, and they are behaving in a way that is not so good for the multicellular organization, not to go into the details. And especially they would produce inflammation, and inflammation would be pro-cancerous. So it would favor cancer in the long run. So it's good in the short run, and it's bad in the long run. So that's the general idea of antagonistic hallmarks. And then there is a third category that they call integrative hallmarks. And they say that these ones result from the first two. Okay, so I'm giving the full picture here just for you to understand what I just said before. In this picture of aging, everything starts with that. So that's the cause of aging. But here what you can see is perturbation, the things that I have distinguished, perturbation and also the cell population dynamics. They are here. And what I'm saying here is that they're not following from that. They are distinct. So they don't come from cell damage. There are other conditions that would help cell damage become a consequence. And on the other hand, or let's say the other side of what I'm saying, is that probably if you looked into hydras, for instance, you would see either perturbation, in fact, both perturbation and cell population dynamics not allowing for damage to accumulate. How exactly? No one knows. But that's obviously what should be happening. Okay, then my last argument. It's already,
No, you're
Charles,
fine.
do you want?
You're fine. No, you can go over it a little bit. Don't worry about it.
Okay.
Yeah, no problem at all.
Okay, thank you. So what I want to say here is that there might be a third way of defining cell aging reductionism. And this would consist in saying, okay, for sure, the general conditions that make senescence possible, okay, I can grant you that they come from non-cellular components. Cell population dynamics and perturbations of multicellularity, or what are sometimes called multicellularity mismatch. The idea that cells are not adapted to living with other cells. So something like this, which is kind of weird, but probably, I mean, not that weird in fact, when you think about it. So someone can tell, okay, I grant you that, but now let's suppose that these two conditions are fixed. They are parameters, they won't change. Okay? They won't vary. What I'm saying now is that most of the variations you would see come from patterns of cell degradation. So that's cell degradation that is explaining everything. So suppose here a cell population dynamics with typical perturbations, and so you have a certain cell population dynamics with cell perturbations and all that I just mentioned, and there are constraints on the further evolution of organisms in a clade. Okay, they all share that, but they have variations. So there are some theories that I can call collectively constraints aging theories. So these are theories that propose this idea that most of aging comes from such constraints. Like the organism has evolved like that, and given that it's like that, it cannot come back on those previous conditions, and that's mostly what explains aging. So these are developed in 2 papers here, and just for publicity, for those of you who are coming to ISH, we are gathering the 2 main authors of those papers, Alan Cohen and David James. They are participating in a workshop with us, a symposium rather, with us at ISH to discuss their theories. Okay, so I don't want to go into the details of examples, but they give a lot of examples of that. Okay. So if we go a little more into the details of such an argument, most of them come or boils down to one theoretical argument that we can call isometry. So here again, suppose that we have very stringent constraints of the cell population dynamics is the same across all organisms in a given clade. It would not change from one species to the other. So no functional variation possible here. What can change is you can have what I would call here isometric time variations here. So what does it mean? It means simply that if you suppose that, for instance, some cell process is going faster, or all cell processes are going faster in one individual cell, in one organism of one species as compared to an organism of another species, what would happen? You would have the cell cycle would also go faster, the cell aging would also go faster, and the turnover of cells would also go faster. And if the turnover of cells in the tissue would go faster, probably organismal aging would also be faster. Okay, you see? So the idea is that if I summarize it very briefly, if the average lifespan of cells inside of the multicellular organism is simply shorter, then probably the lifespan of the organism will also be shorter, if everything else is the same. Okay? So if you think, like, for instance, you have a limited number of cell divisions, of course, if you divide faster, the organism will simply last less long, for sure. So that's what I call the isometric argument. Everything else is the same, the structure is the same, you just change the time. Okay? If it goes faster, then aging would go faster. So now if you, let's say, relax a little bit this very strong supposition, you can suppose that you have little variations in these constraints. Then cell aging reductionism means that most variations would in fact, that you would see, would in fact be variations in cell aging. So there would be variations in this cell cycle. That's the idea, and I think this idea is very, very important in contemporary research. For instance, just to take a typical example, I said, so why on Earth are Hydra not aging? How is that possible? Well, the first hypothesis that comes to most people that are studying Hydra, mostly in Germany, this is where it's mostly studied, what they do is there must be something inside of those cells. They must repair more efficiently. So they look into that, and of course they see variations, and they will say, "Oh, we found the solution. Probably it's because of their, the lamina of it's structured differently. It's more, can bear more damage, or cell repair, or DNA double strand, blah blah blah." We have plenty of mechanisms like this, but the thing is that none of them would explain that there is no damage in the cell. They would not explain why a damaged cell will simply disappear without leaving a trace. They only say, oh, probably a part of the explanation is that the cells are aging less fast. Yes, but they would still age. So we don't have really an explanation. So the alternative would be to say, well, in fact, Hydra are not non-senescent, but this flies against most reasonable evidence we have. Another example of the, and that's a very strong one that you probably know, is that, probably you know that former theories of aging were thinking that the longevity of an organism was proportional to the length of telomeres, which is wrong. We know now is really wrong. I mean, mice have very long telomeres as compared to humans, for instance. But they live, of course, much shorter lives. But one thing that has been observed here more recently is that the rate of telomere shortening seems to scale on the average lifespan. Okay, so that's more interesting. Like the process is a cell process and all that. So you would see that here. In fact, it's very few species, so I cannot really know whether it's not cherry picking here. It's not easy to have that because you have to do some experiments. It's not something you have already in database. It's not. You have to do some experiments, so it's difficult to do that across a huge variety of species. But that's, as you can see, that's an illustration of the same idea. And you can see here, their interpretation of telomere shortening is that it's a loss of cellular viability, which is another way of saying that it's cell aging, in fact. Okay. So I have two arguments against that, but I will summarize them. So this one is hard to summarize. That's too bad because it's very interesting, but... Okay. So some researchers have taken, I will try to summarize it very as efficiently as I can. So some researchers have shown that there is also a correlation between the number of mutations, average number of mutations per year, and longevity. Okay, so that's DNA here, and you can see that, mice have a high rate of mutations. And humans have a very low rate of mutations if you compare. So that's a beautiful curve. You can see it's the same curve, beautiful curve. Okay. But if you read what they say here, it's very interesting because they distinguish that from the idea of cell damage. So you have a very interesting, conflict between two views in that paper. On the one hand, they say, well, in fact, mutations are associated with degradation at the level of the tissue. The tissue is less efficient, the tissue is working less well, but the cells are in fact fitter. Why do they say that? Because they say what they observe is that you have clones of cells with mutations that are more and more prominent in the tissue with time. That's one phenomenon of aging that they have investigated and discovered. Okay, so you will have some mutations would give an advantage to some cells, meaning that you would see these mutations progressively with time more frequently in the tissue. Okay, so that's called clonal clonality, let's say, in the tissue. Usually it's been associated with cancer, but what they show is that in fact it's not cancer. Okay, it's simply aging. For some mutations at least, it's simply aging. Okay. And what is very weird, if you look at how they phrase their theories, it's very awkward, because on the one hand they say that cell fitness is increased, obviously, because these cells are more frequent in the population, in the tissue. But they also mention, well, they say so they are fitter, but at the same time, so the cells are fitter, but the organism, of course, is less fit. Okay, so you have this distinction, and I think that we can very easily interpret that as a case of the perturbation I was mentioning, something like this. Sometimes they say that these cells have damage, DNA damage. But that's weird, because you say DNA damage, but it's inside of the cell, but the cell itself is not damaged, it's fitter. So it's damaged, but it makes the cell fitter. Okay, so that's counterintuitive. I mean, you can understand this. It means that the DNA you have here does not allow, damage in the DNA does not allow the cells to do their job for the multicellular entity, but it's very good for the cell itself, or at least for its propagation, let's say, or this division. Okay. Also, another and second argument comes from the practice of, or the discovery and development of drugs that are called senolytics. So these are drugs that are supposed to rejuvenate the tissue, so improve the overall level of function of an old tissue. And these drugs are simply removing senescent cells that are accumulating in an organism, and in a tissue in particular. And this is rejuvenating the tissue, but it's not rejuvenating the senescent cells themselves, it's simply destroying them. Okay, so here what you have is probably an example of I think, the cell population dynamics. So what I'm calling the cell population dynamics, because it's clearer, is called in that field generally tissue homeostasis. That is the dynamics of renewal of the cells in the tissue. Okay. So conclusion now, because it's way too long. So what's wrong with cell aging reductionisms, if I recap? So remember, cell aging reductionism, the aging of tissues, organs, organisms originates in cell aging. That's my first one. This is how I defined it. Second, I said that for that position, the aging of higher level than cells consists in permanent traces left by cells that have aged. In the multicellular entity, of course. And I have defended cell aging anti-reductionism with three arguments. The first one is that it's not sufficient that cells age for an organism to age. We need also a cell population dynamics. The second one is that it's not necessary that cells degrade for an organism to age. Perturbation is sufficient, and perturbation is not cell degradation. And the third argument is that under some organizational constraints in the multicellular organisms, all variations of organisms aging would not be driven by isometric variations in cell aging. Okay, so what comes next? We can now, if the next stages of my reasoning here would be, so just a few questions to open other further questions, would be, okay, we cannot reduce organismal aging to cell aging. But can we reduce the aging of the organism, to the aging of the tissue in the organism? Maybe we can solve the problem that way. Maybe we can have some kind of tissue reductionism that would hold. Why? Because the tissue is an entity that, in fact, this is where the cell population dynamics makes sense. Okay? But this question would be probably, to some extent, empirical. It would simply be a question of whether cells in one tissue can perturb cells in another tissue. That would be the question. Probably yes. But permanently, this is an open question. Some results, for instance, seem to show that tissues age rather independently from one another, with few exceptions. Okay. A second question is whether the measurement tools we have now to measure the rate of aging, the so-called epigenetic clocks that we use, that are very, very fashionable at the moment, they measure aging in cells. Of course, they can measure aging of the tissue, for instance, or organs or organisms in cells. It's not a problem. But what exactly are they measuring? So that would be the question. And that's the next question I can raise here. And the third one, just to get back to my first question, would be the consequences of what I just said on an evolutionary theory of aging. Because obviously it has some consequences here, I think. Okay, thank you for your attention. That's my team in Bordeaux. Not everyone was there, and as such, you know some people, and there are new faces for you. Yeah, thank you.
Usual practice, quick 5-minute break for coffee and water and restroom if people need it, and we'll be back for questions in just a second. Okay, questions, comments? The floor is open.
Thank you so much for the talk. Super interesting. Super interesting. I do have different questions, but maybe it's just for me a curiosity, a clarification concerning the traces. You know, so the cell engine reductionism. The idea is that we have a lineage of cells, and we have an aging. If I understood correctly, since we have some traces that the previous cell left concerning to the others.
And the organism later, not necessarily the cells in the organism later. It can also be non-cellular components.
Yeah, so, this is the anti-reductionist view.
No, that's also the reductionistic view. The idea is that, what they would say is that the degradation that happens during, so to speak, the lifespan of one cell would leave traces for after it's gone, either in the cell lineage or on other cells or on non-cellular components, whatever. You see, and the point is that any such degradation would always come from the degradation of a cell during its lifespan.
Yeah.
And what I'm saying is that it's not necessarily degradation of a cell that produces that later.
But its clarification is these traces that pass through. What kind of, so sorry, maybe it's a repetition of but it's only really to understand is what do you mean is the mechanical one, the genetic one, and do you think that more broadly, this is a more broad question, what is the role of epigenetics? Where do you put epigenetics, modification, epigenetic marks in all your picture? And specifically also, if there is a link between two traces, can be epigenetic traces, and it's more like clarification for understand better your categorization of them.
Yeah. It's okay. What is So here, one thing is of course that epigenetics appear early in evolution. You would see that, and maybe of course in unicellular organisms. But in the investigation I mentioned in the beginning, one thing that I could establish, at least as a hypothesis, is that you would not see that becoming a mechanism of aging before that stage. That is when you start having multicellular entities, not necessarily multicellular organisms, but multicellular entities like all zones. This is where this mechanism can become an organism of aging. And why? Because you have a trace. Epigenetics can be interpreted in many different ways because it's a very complex phenomenon and that goes in many directions. But one of the effects is that it would modulate the behavior of a cell depending on the rest of the organism. So that's one way a cell that has done something can leave a trace. So if this is what you meant. So I would consider this, epigenetic signatures to be one potential place where you would have such traces, yes. Does that answer your question?
Yeah. So, follow up, if I can. So, this could be also useful, this is a real question, to distinguish between what you mean with damage and what you mean with aging. Because the damage, it seems to me, that is something that do not pass through generation.
Okay, so
I don't know.
It's a very important question. In biogerontology, there is a discussion about the definition of damage now. Because it's not clear. Many people have thought for a long time that it's a kind of self-evident concept. Well, we know when we have damage, and many have discussed that because some say that damage is clear only when it's what they call three-dimensional damage. And what they call three-dimensional damage is basically morphological alteration of some structures. And then they say, okay, but when you have some function that has changed, but you don't see any evidence like the intensity of something, for instance, it's not necessarily associated with a three-dimensional change in the structure of the cell or whatever. So they say damage is probably too broad and encompassing. So I'm not entering that question. What I want to do is simply to rely on the distinction between damage and aging, damage being a broader category, which is kind of consensual. Because if you say every form of damage is aging, you would have paradoxical consequences. So you can very well bite the bullet if you want, but it's probably better if you have a theory of a process that is, again, self-generated and unavoidable. Like it comes from the very functioning of the cell. That's mostly the intuition I think people have. Like there must be something that would happen whatever the environment. Even if you could completely protect an organism from any form of damage, you would see damage accumulating. Okay, so that's the general idea that they have. And then damage is, again, it is what it is, but I think I just need that for my argument, I think.
Okay, thank you.
Yeah. Thanks for the talk. I just wondering how the biological engine is different from the non-living system engine. Like I am an engine now, my computer is an engine now, so I want to curious the difference between.
Okay, so difference between, you said an organism that ages and, I couldn't hear the word you used.
Non-living systems.
Non-living systems. Yeah, so that's an important question. I mean, that's a big philosophical question. So I don't know, but one thing I usually say is that the difference is probably, this is what I said more or less in the beginning of the talk, so I think probably the difference is with the let's say the self-renewal or self-repair mechanism. That would be kind of probably the distinction. But I'm not sure, I mean, this is something that I would probably need to investigate more, because I'm sure that if we can think a little more, there are in nature systems that self-repair and are not alive. So it's not probably a feature that is, let's say, distinctive of organisms. But at the same time, and all organisms have such systems, and they are imperfect in the sense that they can face a certain level of degradation, a certain level of damage or challenge, but not any level. So roughly this is all, I mean, I know it's not satisfactory, but that's roughly what I can say. And there are physical explanations of biological aging as well, in terms of entropy.
Yes, thanks again for the talk. And my question is about the way you define reductionism, because as I see it, you say that how you define multicellular aging is that multicellular organisms age if and only if there is damage in components made in cells, which leaves a cumulative trace. And I was thinking of first, it seems that here it's the fact that there is this cumulative trace which is somehow causally implicated.
Mm-hmm.
And it seems that in the first two arguments you actually directly counter this goal, but in the third argument you do something different, because you say something like, it seems to me that the final conclusion is something like, even if there is something like this cumulative trace, the variation would be not explained by it. So for some reason, other levels themselves are explanatorily relevant for some interesting phenomena, even though if we can still state sort of reductionism in the way that there is still cumulative trace, etc., etc., etc.
Okay, so I'm not sure, but I would say, I'm not sure I completely get your point, but I think that it seems to me that you're identifying cell aging reductionism with cell reductionism, but maybe I'm wrong. Maybe what you're saying here is that it's always cells that are doing, although something in cells would explain the variations, which would be cell reductionism, and I would be okay with that. I think, at least methodologically speaking, I don't have any problem with that. But what I'm saying is that it should be the degradation of cells. That's cell aging reductionism. Cell aging reductionism. It should be the degradation of a cell during its lifetime.
Yes, and I had this second question on the same topic. So you dealt with cell reductionism. Oh, what do you think about molecular reductionism? Can you say something like there is aging only if there is accumulation of damage at molecular level which somehow leaves cumulative trace? Because I see that, for example, in the second argument you presented, it still will hold against this because it's very general consideration that cell may perturb something at a high level, but many of the examples you gave as sort of counterexamples of potential cases where we see this non-reducible to cell degradation aging, there is still something like DNA damage.
Molecular and So one thing is for sure. I think it's an important point. I should probably make clear in further versions of the argument. So first, in the literature, you would find some people saying that it's consensual that the damage of an organism is about the damage of the cells in that organism, and many would say that it's reducible to cellular and molecular damage. So they would say that. But the problem, I think, why many people shy away from saying that is that it's very weird for many people to say that a protein is aging, for instance. Mm-hmm. So they would say, you cannot really say that. So that's probably the reason why, I think. And that said, I would think, and that's why I'm focusing more on cell aging reductionism, that most forms of molecular damage you would find inside of a cell would also disappear with the cell itself. So there's an argument about whether some damage inside of the cell can leave a trace outside of the cell. So which it does, of course, but only if it changes the behavior of the cell itself. So like a mutation can, of course, have an impact on the tissue, provided that the behavior of the cell is changed within the tissue with that damage, for instance. Mm-hmm. So in a way, it's a kind of shield. The cell is shielding the tissue from effects of molecular damage inside. But then there would be a second part of your argument, I think, that would be about molecular damage outside of the cell, which is the case for the ECM. And there, I think it's an interesting question. And as you know, Jean-François Moreau, our colleague in Bordeaux, is developing that idea that this is the main cause of aging. The one and only cause of aging is the degradation of the extracellular matrix, according to him. So he has a very strong argument. I'm not completely buying the argument. I think it's a bit probably insufficient, but I think it's a major point he's making. So it's called, in the literature, it's called fibroaging. And there are some people that are trying to measure the signature of an age ECM.
Thank you, Maël, for the talk. I have a few questions to better understand. Mm-hmm. You say that some population dynamics is necessary, and a way for me to help me to understand, because at the end you give a specific case, but you don't qualify the kind of dynamics that would be necessary. Mm-hmm. I would like more theoretical. But the idea, if I understand correctly, is that even if we have an organism with immortal cells, it could age because of that dynamics.
Well, no. If you had an organism with immortal cells, it could age if perturbations would happen.
Yeah, exactly. It's a certain kind of dynamics among the cells, blah blah blah, aging.
And and especially the idea of perturbation is that this dynamic would be transformed. So originally it could be perpetual, but the cells are doing something that would have this dynamics shift to another dynamics that cannot be perpetual.
Okay. So you cache it as perturbation, but could
Yes.
you say more? Because it seems to me that, systemic people, system biology would say, oh yeah, we can qualify that kind of thing. So, or is it too speculative?
No, I think it's very interesting, and this is me and my own, let's say, shortcomings. I don't know how to make that more precise as of now. This is something I would very much like to develop, but I'm not competent enough to do that. And I think I'm not sure we have the data though, except for some phenomena like clonality. There we have some data, and this is me not knowing enough of that. So that's why I'm a bit. evasive on that question. So I just.
But you see a general mechanism. At least you.
I mean,
seems to agree.
I think you saw that in the asymmetric division. I mean, it's a very simple case, but you can see that can be generalized. I think you can see that it's that there you would have that problem generalized. So I think it's really about the distribution of damage across all lineages.
Can I?
Actually, can I let myself in with a follow-up? Because I have a very similar question about the next argument. So you slip this in near the end, and I wonder if it's something you want to go all the way in on.
Okay.
Similarly, is the general characteristic of the second set of examples, are they all mismatches of some sort or another? Because you dropped in mismatch, but as you were describing the examples, I was like, well, this is kind of a weird kind of mismatch kind of thing, right? Do you think that's going to work? Is that going to hold, you think?
I don't know. I mean, I could withdraw the term mismatch. I'm not sure about it. I mean, I know that it has many connotations on top of that, and it's not an orthodox use of mismatch. But the idea basically is that a cell is not perfectly adapted to its multicellular environment.
Because I was getting the vibe as the examples were being introduced. I had independently written in my notes, like, is this something like mismatch? Before you said it later on at the end.
Yeah.
So I mean, I get what the intuition is. Yeah, I think it's interesting, the question whether or not that's sort of the common characteristic that unites those examples or not.
Good point. Okay,
I don't know. I thought it was kind of a cool provocation.
Yeah, I like it. I mean, it can be stated in various ways, and I think the simplest is probably to say that, as I tried to say, that we should not presuppose that everything that normal cells do is good for the even in a healthy organism is good for the organism. They're just doing their stuff, and it has to be sufficiently, good for the preservation of the multicellular entity. But it doesn't need to be all so that when they do something normally, it could be bad for the multicellular organism without any disease or degradation or whatever. So that's the basic idea. So that you don't have a perfect adaptation of cells inside of multicellular organisms, which is maybe counterintuitive, a little bit provocative, but when you come to think of it, it would be pretty, I think, it's not that controversial, I think, to say such a thing. But I would need more examples, obviously. Yeah. And also we would need to think more about mismatch.
Yeah. Okay, back to you. Sorry.
About the argument about the asymmetry of damage, I don't understand how it works, because to me this argument will show that, okay, there's a possibility with damage to have a maintaining a population of cell, but not to grow. The vice point argument is that if you have damage, or you all die, or you don't grow, you cannot grow in number in the population. If you transfer all the damage to one or the two, you divide two and you get one. So how does it work?
Because I mean it's not an all or nothing thing. In fact, what happens is that when you have one cell that is dividing into two cells, and you have one, say, that is damaged.
More damage.
More damage than the other. So this one would give rise to the same thing with the same level of damage and one new one, while the other one would give rise to one with more damage and the other one new. So in fact, this generation, you have already, you had one here, you have already two perfectly clear ones. So in that case, you would have exponential growth, except that it's not, it's not as fast as it would be if the two were perfect. But you can have a growth. Is it clearer?
Okay.
Yeah. So what I didn't say is that, of course, it's not like one of them would die without reproducing. It simply would take more time to reproduce, and then you would have even more damage.
Okay.
So I didn't explain that the mechanism is accumulation of misshaped proteins.
Okay.
So it's a wonderful example, because what happens is that when proteins are misshaped, they would have a tendency to aggregate. And given that all of them would aggregate, they would aggregate into one body, and not 2 or 3 or 4 or 5 or whatever. So if it's one body, given that cells divide, the body would go either in one or the other, but it cannot, divide itself. So and in fact, this is what they studied. Statistically speaking, it's quite rare, but it happens sometimes that you have kind of asymmetric because you have two bodies. You have a smaller one and a bigger one. And then the small one can go in one cell and the big one goes in the other. But in 90% of cases, you would have that inclusion body only in one of the two daughter cells. So that's wonderful. It's just chemistry.
It's Stewart paper? Or?
Yeah, that's the Stewart paper. So it's really cool. I mean, you have a very elegant explanation of how and why unicellular organisms don't disappear or haven't disappeared.
Good, thank you.
It's just a chemistry. I mean, you have the same example with sexual reproduction, in fact. I didn't mention it, but it's basically probably a repair mechanism. And so this is what organisms that would accumulate some damage with time can rejuvenate through, for instance, the ciliates do that through conjugation. So they conjugate, so they have a kind of mating process, and then so the two would fuse into one, and then they would divide again into two, and both would be repaired. Like they use each other's DNA information to repair each other. So that's wonderful. And you cannot call that reproduction because you had two organisms in the first place, and you have two organisms again in the.
Good, thank you.
Sorry.
Yeah.
Other questions? No, it's a little bit a follow-up on the first question, and Charles follow concerning the perturbation, because I had in mind the same question, because it seems what is the difference then between variations and perturbations? Is only because we can have also cell-to-cell variations, and is perturbation only a normative declination, can I say, of the variation? A perturbation is a bad variation. This is my first point.
Okay, can I try to answer that one?
Yes, sorry.
Yeah. So I would probably think that it's sufficient to say that perturbation simply is a variation that has a consequence on the long-term preservation of the population of cells.
Okay,
Of the tissue. Yeah, so that way I think we can say that it's not normative. It's not like I'm saying that these are the variations that are bad, while those are good. You can say, well, I'm not saying whether it's bad or good, I'm just saying that these ones are the ones that would lead to non-perpetual and to non-perpetuity, if you want. And these ones can lead to perpetuity.
The second point, I don't know if it's relevant enough for me, is kind of all this debate about perturbation make me think about stochasticity in cell, and the debate between what part of stochasticity can be functional and which part can be only detrimental. It's something. It has something to do with your picture, or is something more related? I don't know. Because I cannot see the links, but I think maybe between perturbation and stochasticity and noise in gene expression, also the problematic, or at least the debate between stochasticity versus noise and the role of stochasticity in cells or between cell-cell communication is something very debated in biological literature today, even if now stochasticity gain more attention concerning its possible functionality. And I was wondering concerning the aging topic, what is the place of this debate, this if any?
It's a very good question. So there are stochastic theories of aging.
Okay.
So entropy, let's say several variants of an entropy theory of aging. But I mean, if you know of Schrödinger's essay, What is Life? And what he says there is that entropy cannot be a direct explanation of the aging of organisms, simply because an organism is defined by what he calls negative entropy. It says that it's a property of an organism to be able to replace internal entropy or exchange internal entropy with external entropy. Like get rid of the entropy by putting it outside of the organism. So that's his idea. And so everyone knows that argument, but nonetheless, many people say, yeah, but the first primary cause of aging must be stochasticity somehow. And what I tend to think is that this condition is in fact universal and invariable. So it cannot explain variations. So either all organisms would age or some of them only do. So if some of them do, given that all of them are submitted to entropy, it means that the explanatory factor cannot be entropy. So that's my view on the thing. That said, I have to say that there are very interesting theories that are developed nonetheless, and I have not yet had the time to investigate them, with all the details. And what I would say is that probably if you look again at that figure here, I think that the stochasticity would probably explain why you have some damage, but let's say the non-senescence of some organisms would anyway show that you can, even though you have stochasticity, you can have perpetuity. So there must be a second factor. So it has to be, if stochasticity is one necessary condition, there must be another one. And this other one is somewhere here. And I think, for instance, you would have stochasticity here in that tree because, as I said, asymmetric damage distribution need not be, always an all or nothing thing. It can be a 80%, 10%, it can be, 80-20, or it can vary. And in some cases, it would never be, like if you are closer to 50-50, for instance, then probably you would have an organism that is much more likely to degrade. So, and that kind of stochastic effect is studied, but it's very difficult to investigate, with a certain level of certainty at the level of stem cells, how they divide. So that's a very interesting, important question. Asymmetrically, symmetrically, there are many models for that. And I think it's very key to understanding the organism and the aging of tissues. So yeah, you're right, it's a very important question.
Yeah, but at the end of the day, it could be, stochasticity could give us factors among others.
Yes.
Yeah, okay.
Yeah, and I would agree with that. I think it must play a role somehow.
Yeah.
And there's also, it's for sure, organisms that are non-senescent, it's also a mistake that is sometimes made. It doesn't mean that they are immortal. They can die, of course. They can die in a nuclear explosion or whatever, but they can also die if the level of damage is too high for them to repair, which is very interesting. You see that in sponges. Sponges, if they have a certain level of damage, they can perfectly repair and they can live thousands of years. But if they have also a rate of repair or proliferation, if the rate of damage is higher, they will degrade and die. So, there's no such thing as an immortal organism. So non-senescence is not about immortality, and that also is stochastic somehow.
Nonetheless, there is one line of cell that is called HILA that is sometimes called immortal cells, and
Yeah.
it's very used in the research as a cancer.
Still, if under a nuclear bomb.
Yeah. Yeah. That's interesting as well. It's I don't know very well. I know that, but I haven't investigated that. The case I would have investigated is rather the case of the Hydra cell. This one is very puzzling as well. But you're right. It's a good suggestion. I should look more into what that means, that it is immortal.
Yeah.
Yeah. And when we say cancer cells are immortal, for instance, that's a huge approximation. They are not immortal. They're very messy, and at the level of the tumor, yes, it looks like they are immortal because they grow out of, but it doesn't mean that they would grow, eternally and all that, because they accumulate damage and they are more and more damaged, more and more aberrant cells. So it's not true that that would involve that they are stable, which is not the case at all.
Thank you. Just to continue the point about Schrödinger, because of course thermodynamics evolve after, and people will say, okay, I will try to sell Belgium again. So of course, Prigogine, non-equilibrium. So from the point of view of thermodynamics, an organism and a hurricane are similar. But of course, as you say, it's in the variation that you see the important point. It's not the entropy bullshit, because entropy is a general concept that is not that useful at that level. However, the mechanism that maintains the system out of equilibrium, if it's damaged, that's aging.
Yes.
You could say that. But of course, after that, it's how to qualify this mechanism. Because in a hurricane, pretty simple, not living very old.
Yes.
In an organism, we can imagine very long time. So
Yes.
how the mechanism could be damaged to the point that you cannot maintain your system out of equilibrium, and that could be not related to cells at all. Because
Exactly.
that's a property of the system.
Of the system. I agree.
So maybe it's the cells, maybe it's the organization, maybe it's something else, but I don't see why the cell reduction answer seems, it's like someone saying to me, okay, the problem with why house become not house, it's the bricks are disappearing.
Uh-huh.
And okay, of course the bricks erodes, but usually there's systemic something breaking before the bricks. So why, my question was why the cell hypothesis, cell reduction, aging hypothesis seems so
Prominent.
prominent.
Yeah. So the first, I should say 3 quick things about that. The first one is that the field is moving in a direction that is less cell reductionistic.
Okay.
So that's obvious if you compare Lopez-Otin 2013 to Lopez-Otin 2023. Whereas it's very interesting because they have removed all of the sentences that were clearly cell reductionistic, and they have kept the framework generally. So you have kind of
Quick aside, they just rewrote their paper 10 years later.
Yeah.
Because they were like, we need to do a new perspective on
exactly,
where the
and
field
there are
is.
new. In fact, the
Okay,
point is
okay,
there
cool.
are new mechanisms that we have discovered.
That's actually kind of cool. Yeah,
Yeah,
okay.
and it's the same. That's the same with the hallmark of aging paper. Every 10 years or so, they rewrite it, and most of the time, what happens is that you have a very clear theory in the beginning, and you have a very confusing theory in the end. It's not a theory; it's a list, basically, because the theory cannot hold anymore. It doesn't hold anymore. So that would be the first answer. Second answer. I think that it's important, evolutionarily speaking, because the question is where and when exactly. Yeah, it started. And I think it's a question of level. As I was saying, I think what we want to study is metacellular aging, something that happens in the organization. So you take it for granted, and I agree with you that this should be the case. Everyone should say, well, we're interested in the aging of organism. So it can be the cell, it can be something else. It doesn't need to be the cell. But many people say, wow, that's in fact a sin that is was inherited from unicellular organisms down to, so something like this. And so they have this argument as well. And the third reason, which is also pragmatically very important, is that most researchers that are trying to find ways of, putting you on the right track of aging, like you have a good, trajectory of aging, they try to intervene at the level of cells and on cell aging. So some of them do that. Others, and with the senolytics, it's kind of evident that in fact, this is not what they're doing. They're trying to remove, as I said, senescent cells from the tissue, which is not the same thing as rejuvenating cells. But in other cases, they try to rejuvenate cells. So it's also a question of strategy. Where should we intervene? At the level of cells or at the level of the organization of the tissue? So there's also that kind of pragmatic question of which strategy, which therapeutic strategy is best.
Okay.
But I completely agree with you. It's kind of obvious. So if you summarize it like this, you would say my point is really very trivial. I think it is trivial, but for the field, it's not, because they have all these biases. But for sure. And then the next question, as you said, is the next important question is to have a more detailed description of the mechanisms. Why is that system degrading with time, and how? So that's and to answer that question, you have to go into the hallmarks themselves and see how they interact exactly. So this is why it's a very preliminary draft of a theory of how a system.
But Maël, I didn't want to say it was trivial. Okay? It's trivial for houses, but organisms are complicated.
Yeah, but I think it's. This is me saying, I know it's not you, it's me saying that it's a trivial point. Because I could just explain the view as you just did. I could say, just like in a house, it can be this or that, it can be bricks or something else. And I think everyone would say, yeah, of course. What of that? So the points, this is why I was developing the other points. But I think it could be said like this, yes.
And the theology building informs us that unfortunately our time is up. So let's thank our speaker.